And the logistics out of the way, I'm going to hand this over to Lisa, who will do a little bit of an overview and introductions. Thank you.
Thanks, Joe. So Courtney, if you pop to the next slide. So we talked about this on Tuesday, but just to orient everyone, we've started this seminar series through the ACTIV Trace Group, and I co-chair the ACTIV Trace Group with NIH and NIAID with Matt. And we decided to move forward and focus on effector function only because it's a field where the experts know a lot about effector function, but outside of that expertise, we really don't have broad knowledge around how antibodies, whether induced by a vaccine or from monoclonals, how they work through their FC. And so because there really is not much attention on understanding how this works, we had decided to put together a couple groups, and they focused on different aspects of what we thought was pretty important in the field of effector function. So maybe if we go to the next slide, I think this outlines the groups. We had two groups, one that really focused on and are focusing on the standardization of effector function assays. So one of the things is that we all use very different assays to describe and understand effector function, whether that's from sera or from monoclonals or whatever you're sort of looking at complement. And this group was headed up by Annie Zumsteg at Veer. We had a number of participants, all experts in the field: Marty Galit and Arvin Stelios from Rockefeller, Davide from Veer, as well as Mike Diamond, Stacey Ferguson, Mikkel, who we heard from on Tuesday, Georgia, Tea, and Jerry Weir. And then the second group was focused on translation of preclinical effector function to the clinical COVID experience. This group, which had headed up the seminar series, was led by Galit and had Ann Stelios, Jim, Mark Esser, Stacey Ferguson, Nicole Calloward from Lilly, Jerry Weir, as well as Annie, and thought that in order for us to really gain an understanding on how to translate, that we really should sort of set the stage on effector function, and therefore really wanted this seminar series to move forward. So if we go to the next slide. So, right, just to mention it before we get started, the next presentation is from Arturo, and that's on the 21st of December. So we've got a couple weeks before that, but he's really going to look at convalescent plasma correlates of protection. But if we go to the next slide, what we're talking about today is from Jim. Jim's going to present on effector cells in different compartments and disease states, which should be a pretty robust presentation. Just to introduce Jim, although he probably needs no introduction at all. He's the director of the Vanderbilt Vaccine Center and founded ID Biologics, and really focus on human monoclonal antibodies and a huge portfolio of therapeutics and leads within that company. He's also professor of pediatrics, micro, and immunology at Vanderbilt. He's actually a board certified pediatric infectious disease specialist, received his MD from Chapel Hill, did his postdoc in NIH virology, in immunology and vaccines. And like I mentioned, Jim's a powerhouse in viral immunology, discovered many vaccine candidates, monoclonal antibodies, ones that are now in the clinic or approved. Some of you may have heard of Evishield, that came from Jim's lab. Ongoing work in any number of viral families, you name it. I'm sure Jim has studied it and has a monoclonal for it. Ebola to Staph aureus, to toxins, to rhinovirus, including many others. I was going to list the awards that Jim's received, but I'm pretty sure I would take us right all the way to 11 a.m. Pacific time if I did that for the next hour. But many awards. I feel like every time I log into LinkedIn, there's a new one that Jim's received. March of Dimes, American Society for Microbiology, Pediatric Infectious Disease Society. He's an elected member of the National Academy of Medicine and National Academy of Inventors, elected fellow of many other organizations. So without further ado, I'm going to pass it over to Jim. I'm really looking forward to your seminar, Jim, and looking forward to the questions and discussions at the end. So take it away, Jim.
Okay, thank you very much, Lisa. That was generous. Yeah, you just burned some of my time there. But thank you for that kind introduction. I'm trying to find my, so I can project this. Now I'm good. All right.
Yeah, I think it's, there we go.
Yeah, so I'm gonna try to think through with us today how do antibodies actually work and what's the best way to use them. And I'm going to focus more on the desirable properties of a drug development view than only characterizing mechanisms of action, say in natural immunity or vaccines. I'm going to transition all the way to the idea of what makes a good drug. I think it's ironic that in the field of human monoclonal antibodies or monoclonal antibodies in general, there are over 100 that have been licensed by the FDA and or the EMA, but they're not for infectious diseases. They're almost all for cancer and autoimmunity, which is weird because if you look back at the history of antibody science, started in the late 19th century, and people were immunizing horses and bleeding them and making serum, and that horse serum was used for rabies and for tetanus and diphtheria, especially in World War I, became a major application. And so we knew antibodies were important for infectious diseases. That's the beginning of antibody science. In fact, even before we knew antibodies were antibodies, toxins like diphtheria and tetanus were being put into animals. So people conceived of these molecules as antitoxins, and that's where the word antibody eventually derived from. It was antitoxins, and then if the targets were more diverse than toxins. Of course, Emil von Behring, there's a lot of work going on in the late 19th century, but Emil von Behring got the first Nobel Prize for antibodies, actually. And there have been many Nobel Prizes in the area of antibodies. I think in infectious diseases, antibodies have not been particularly important other than as polyclonal antibodies in the 1950s and 60s. Then we transitioned to the vaccine era. But I want to make the point that I think antibodies really have potential to serve as vaccines or to outperform vaccines and antiviral small molecule drugs for a lot of desirable reasons. They have not yet achieved that, but I think the future will include this. So I think in the future, people won't be saying, oh, we should just always have vaccines. They'll say, well, one of the go-to strategies should be to get antibodies. And that'll be sort of our left hand and right hand, and I'll come back to that. Antibodies are interesting because they can be used, and we've just seen really the first proof of principle at large scale of using them as vaccines for pre-exposure prophylaxis. Historically, prevention has been the sole province of vaccines. We have not so much used antibodies other than travelers for hepatitis B historically and Hep A and so on, and perinatal exposure to maternal Hep B we still give, or rabies. Post-exposure prophylaxis is another example. Hep B and rabies, we're using antibodies for post-exposure prophylaxis. But also antibodies could be used for mild outpatient disease. We've seen that in COVID. That's typically not attempted in any format other than some flu drugs, which don't work very well because it's usually too late. And then finally, treatment of severe diseases, usually the target of small molecule viral inhibitors, but they really only work well in chronic infections. They're not particularly good for acute infections, but antibodies have been used successfully in all of these. So if you think about it, it's amazing. You got a single molecule that can be used for prevention, post-exposure prophylaxis, treatment of mild disease or severe disease. And that's why I think they have so much upside potential. I'm surprised that this group invited me to come talk on FC effector functions as being a predominant thing. I really am sort of an outlier. I don't believe, and I'm just going to try to convince you the scientific basis for this. I don't believe that typically FC-mediated effector functions are the predominant mode by which antiviral antibodies mediate immunity. I think they're an adjunctive activity that you sometimes want and you sometimes don't want, but you need to think about that thoughtfully. But over and above that, typically neutralization is going to be the driving property of antibodies that mediate antiviral antibody function. And when you look at the field over the last, say, 30, 40 years, typically in vitro neutralization with authentic viruses or sometimes pseudoviruses, et cetera, predicts in vivo efficacy and ultimately clinical effectiveness. And I know many people on this call will be not agree with that statement and say, well, hey, look at my paper. I saw some FC effector function that correlated with, but my meta-analysis of the field is that neutralization is the primary property that you're seeking. And when you don't have potent neutralization, then you start seeing a more significant role for FC-mediated activities in antiviral. I'm not talking about cancer when you have to eliminate cancer cells. I'm talking about viruses, antiviral immunity. Now, within, what devolves from neutralization as the major predictive property is that the most potently neutralizing antibodies typically are those that bind the receptor binding domain and block attachment. So if you can block attachment, it's sort of game over for viruses, and that's the best way to prevent virus infection, to block attachment to cells. Now, there are antibodies that don't block attachment. They block fusion, they block conformational transitions, they block egress. There's all sorts of mechanisms of activity of antibodies, including multiple. There's probably a dozen molecular mechanisms of neutralization, but I'm just saying, if you look across dozens and dozens of viruses, typically the most potent neutralization activity comes from blocking attachment, which is blocking receptor binding domain typically. And okay, so I'm boosting antibodies here, but I'm also cautioning it's likely that if you throw antibodies in late during infection, particularly in mucosal diseases like influenza, RSV, para-3 infections, COVID, pulmonary infections, if once you have a full burning fire at a mucosal site and you throw in antibodies that tag or they mediate molecular recognition of infected cells, and those antibodies recruit complement deposition and other immune effectors to come in to spray cytokines left and right, and CTLs kick in, it's likely that antibodies contribute to inflammation when they're used during active infection. So therefore, antibodies are not always, quote, good. I think we need to use them thoughtfully, especially during active infection. And those sort of complement deposition and inflammatory acceleration or contributions are mediated by Fc receptors. So I think that's something we need to keep in mind. Now, there has been, I think, historically kind of a balance between just pure neutralization and these other effector things where complement or ADCC or ADMP and all the other things that are being explored in this group by other speakers. But the reason I think not only neutralization typically is, quote, better if you can achieve that, the potency of antibodies that we can discover now is just amazing. We've moved multiple regimes, multiple logarithmic steps in terms of the potency of antibodies that we can get for neutralization. And if you can get an antibody that's very ultra potent, then you probably don't need any other effector functions, especially if they're attachment blocking. And I'll just give you one example. This is partially published and unpublished. We've been working on Rift Valley fever virus, which is a Bunya virus that causes hemorrhagic fever syndromes in East Africa. It's a One Health target because it gets sheep and goats, a lot of sort of cattle scenarios, but then the farmers get it, and then you can have human infections that are terrible. It causes a lot of trouble in pregnant animals and pregnant women. So it's a bad virus. It's a good target to work on. One of the graduate students in our lab, just a single graduate student, did some discovery campaigns, Nate Chapman, who's actually now in at Duke with Georgia Tamara. I was working on FC effector functions. So it's pretty interesting that Nate Chapman, who did this work, is working on FC effector functions right now. But he did a campaign, and these are just a few of the antibodies that were the potency. So the antibodies are listed in the left column. The glycoprotein of bunyaviruses is complicated. There's a GN and GC that come together. The antigenic target of most of these antibodies with the GN component. And if you look at the IC50 value, or the half maximal effective value for neutralization, we're getting antibodies that are lower than 1 nanogram per ml IC50. So this type of potency gives you a drug that's more potent than most small molecule drugs ever are seen to be, and sort of unprecedented activity. And so what does that translate into in vivo? So we've been doing studies in mouse models with Rift and rat models with Amy Hartman and Pitt. This is work that was done at Utah State with Brian Gowan and John Westover. So if you give a fatal inoculum to a BALB/c mouse, and we've done prevention or post-exposure, but this is actually a treatment when the mice are already symptomatic. And this is an unpublished study where we were doing a dose down, 22 micrograms per mouse, 2.2, 0.22, 0.022 micrograms, and then 0.0022, which is on a per kilogram basis, 0.011 milligrams per kilogram. And we see protection against fatal inoculation all the way down to doses as low, partial protection, 0.01 milligrams per kilogram. So just to calculate that for you, that's 11 micrograms per kilogram, which in a human is less than 1 milligram. So in flu challenge studies or in COVID, human beings have been given as much as 8 grams of antibody, and I'm talking about less than 1 milligram is predicted to be effective for a 70 kilogram person. So the potency of these antibodies and the in vivo efficacy is extraordinary and unprecedented. We've never seen antibodies that are this potent. So this changes the rules around what neutralization does versus other activities and what's desirable. And you could say, well, wow, that's just kind of a one-off. How commonly would you be able to do this? Or why is that happening all of a sudden? Why are we able to find ultra potent antibodies that are less than 1 nanogram per ml IC50 all of a sudden? Well, that's because the technologies that we're using in discovery are moving so fast, you can't believe it. So this is an experiment. Again, these are unpublished data that hopefully will be published soon. We took blood from an Ebola survivor from the West African outbreak in 2014, isolated PBMCs, did B cell, and then labeled B cells with Ebola GP, the surface glycoprotein, and then did single cell 10x genomics work. And this is, again, a single graduate student. This is not a company environment. This is just one graduate student isolated by flow cytometry, identified 120,000 on flow, 120,000 virus-specific or GP-specific cells, and we recovered the heavy and the light chain for 15,000 of those from one specimen. And if you look on this figure on D, species richness, as you go from right to left with the 15,000, how many unique antibodies do you see? And the purple, gray is infinite, and the purple is what we saw. And what this tells us is we keep finding new antibodies, even when we get out to 15,000 clones in one specimen. This is just one person at one time point. So the diversity of the human immunome around a particular infection, this is just one protein, is enormous. And we predict that individuals have tens of thousands or more individual clones to a virus like Ebola GP. And so we know the sequences of all these because these were done with RNA-seq. And so we start doing clustering and say, well, are they all perfectly unique, or are they related into lineages or clonal families? And so if we start putting the 15,000 out, we can see, yeah, they fall into clonal lineages. And this, there's a lot of colors on this. This is an infographic. So the orange ones are broadly reactive. So they'll get Bundabujo, Zaire, and Sudan, which is highly desirable, lots of species. And the purple and blue ones are more species specific. So you can see the clusters of antibodies in this person. Some of them are very broad. Some of them are species specific. So there's a lot of phenotypes but anything you want is present in this person's repertoire. And so we went and did Zaire, Bunabujo, Sudan, and even Marburg binding and neutralization with authentic viruses, and looked at reactivity to cleaved or uncleaved proteins. And so we did a lot of work on this to phenotype these antibodies. So that's all done with single cell techniques. Now, Lisa mentioned the compartments. So when we're doing single cell techniques with memory B cells, you're looking at blood typically. Sometimes we use bone marrow, sometimes we use lymph nodes, sometimes we use gut biopsies, but typically it's memory B cells in blood. Other groups, and sometimes we use plasmablasts that occur on approximately day 7 after immunization or infection. But typically people are doing memory B cell work. But the antibodies that protect you against re-exposure, reinfection are coming from long-lived plasma cells in the bone marrow. So in this individual, not only did we make 15,000 antibodies from DNA, we collected plasma and captured protein antibodies from plasma onto protein GP and eluted those antibody proteins and sequenced them by mass spec. So on the left, you see the protein sequences we got. We got thousands of heavy and light chains, and then look for the overlap between the memory B cell compartment and the long-lived plasma cell, i.e. the plasma IgGs. And you see lots of overlap there, and even lots of clonal families. So it's interesting that the clones that go into memory B cell, they may or may not be fully represented in the bone marrow, but many of the ones that end up in the bone marrow that going to mediate your protection are in both compartments. And I mentioned how many clones there are, but if you look at each clonal lineage, these are just 3 representative clonal lineages, and the colored bars left to right are monoclonals that we recovered. But if you look in the deep sequencing for this individual, the left is the heavy chain and the right is the light chain. These are somatic variants. So even for one particular monoclonal within an individual, you can see hundreds of somatic variants that may have different breadth or reactivity. And then what's interesting, if you take that individual that we looked at, which is the top right of the plot, and other individuals from the outbreak, and compare those to information in the literature, we had more sequences than the entire Ebola literature from this one individual. But that allowed us to find what are called public clonotypes by aligning our sequences to published sequences from VSV vaccinees or adenovirus vaccinees or other survivors that were treated at Emory. From the Emory group has published a lot of work, Scott Boyd and others. And the connections in this circus plot are sharing. So you can see individuals share antibodies between each other, and you can then overlay all the phenotypes of all these public clonotypes. And I don't have time to work through all this, but basically you can see species specific or broad and neutralizing and non-neutralizing in public clonotypes. So there are a lot of shared clones that people make to Ebola that are neutralizing and even broad. And we went on to do structures of all these. So these are the top 15 public clonotypes, and they decorate the GP from the very top, the side, the bottom. So not only is there genetic diversity and breadth diversity and potency diversity, but this single individual is decorating the entire GP. So the breadth and neutralization in one individual is probably sufficient. And then does that correlate with in vivo? We work with Alex Bakreyev at UTMB, and he put the top 15 public clonotypes that were shared among individuals into mice, and most of them are fully protective as monotherapy for Ebola. So that's kind of why we're finding these antibodies that are so potent, because the technologies are allowing us to make thousands and thousands of antibodies, and we can sort through them. And stochastically, you're going to find some that recognize conserved regions and are very potent, and that's very desirable. Now, I mentioned at the beginning, most antibodies that have been licensed are for autoimmunity and cancer or cholesterol, all kinds of crazy stuff, not for infectious disease. Prior to COVID, there was just a few antibodies that had made it for infectious disease. Of course, Synagis or palivizumab since 1998. We've seen some HIV, an HIV antibody, and then Ebola was really the first case where you started seeing expansion, where you either have a cocktail of 3 or monotherapy made it to approval, anthrax antibody. But during COVID, we just saw an explosion, and this is sort of a beta test of what you can do with antibodies for infectious diseases. One of the points I want to emphasize, again, I mentioned antibodies can be used as vaccines. And if you wrote what would be called a target product profile for a vaccine or a treatment, because those are different, right? Target product profile for preventing or treatment. Prevention might be, hey, I'd like to have 80% protection against all disease, not even just severe disease. And I'd like to be able to prevent in high risk and immunocompromised people who are at the highest risk. And then for treatment, I'd like to give 1 IM dose and be done with it. And 90% of people be protected from progression to severe disease. And in fact, this is not a hypothetical. This is actually what happened with Evusheld that AstraZeneca developed based on antibodies Lisa mentioned we discovered in the lab here. So the Provent trial, this was it, 83% against all symptoms, not just severe, but even URI, which far exceeds the efficacy of any of the mRNA vaccines at 6 months. And I think you're probably going to say, what? That can't be right. But you go back and look at the efficacy trials of the mRNA vaccines. The reason people I've gotten five doses of mRNA vaccine myself is because the durability is quite short. It is not as good as these antibodies were in high-risk patients. And then in the post-exposure prophylaxis, you also see the same molecule working. Now, just briefly, where did these come from? We made SARS-CoV-2, the same sort of thing I just showed you. Take blood, memory B cells, antigen-specific sorting, go to either expansion or single-cell RNA. We did this with either 10x Genomics or Berkeley Lights Beacon, and then you end up with genes. Those genes go into CHO cells. Now you have recombinant antibodies, and you do binding and neutralization. And the Beacon for us was really helpful. It was the first time we ever used it, but it's a pretty interesting device where you can do single-cell stuff at high scale. So we covered beads with SARS-CoV-2 spike protein, and each individual cell goes into these little wells that they call pens, and the antibody secretes out into a channel. And so you can identify individual cells that are alive. This is a movie I'm showing you of a couple hundred of the pens. I think there's about 18,000 of these pens on every chip, but this is a few hundred. And this movie, I think, is 30 minutes. So if you watch over the 30 minutes, an individual cell at the bottom of a pen spews out antibody like a volcano flume, and it decorates the protein beads. And so you go in and you essentially export, you grab each individual cell and put it into a collection channel, and then you essentially get that genes out of that one B cell. And you just do this all day long, particularly if you've pre-selected antibodies that have a phenotype of interest. So we go from PBMCs to either 10x Genomics, single cell, or Berkeley Lights. Then you sequence everything. We worked in particular with Twist. So I call it emailing, but you basically FTP the sequences onto their server, and they synthesize the DNA and ship it back to you, and you make thousands of antibodies. We also, since I already told you my cognitive bias, I believe neutralization is the driving parameter to look at. So we developed real-time high throughput virus neutralization assays that can be done in 384 well plates, where you can see cells filling in and making impedance on a cell impedance device. And if there's no antibody, the virus kills the cells and you get full CPE, and the cells blow up and you lose your cell index or impedance. But if you add virus and antibody, the antibody neutralizes, the cells stay up and they're fine. And so we actually did dilutions. And so live fire with COVID, we developed a test, and you can do it with pseudovirus or real virus, and even did dilutions and screened with IC50 with neutralization for thousands of antibodies. So that's how we did it. Another important concept in the field is using 2 antibodies. So we're always going to use 2 antibodies or more for RNA viruses in future, most likely because of the risk of variants and escape. But there's value add if you don't just pick 2 antibodies randomly, but if you sort through the thousands of antibodies you get and add one and the other very carefully in dilutions, you can build what's called a synergy map. And you can identify combinations, which are not the usual, but rare combinations will be cooperative or synergistic. And this turned out to be very important for Evusheld in the clinic, because sometimes one would go down, but still synergize the other if you didn't see loss of binding. This is a single particle EM of the two antibodies that ended up in Nevis-Sheld on spike. And we ultimately did, crystallography, and this is needed for the FDA to show mechanism of action. But two antibodies combined very closely on an antigen and not interfere, in fact, synergize with each other. Another important concept in the neutralization paradigm is difficult to escape. So we worked with Jesse Bloom, who put every amino acid at every position in the spike RBD and displayed it in yeast. And then we were looking at antibodies where single nucleotide substitutions were required to escape. And these web logos show you amino acids that mediate escape. And for these particular antibodies that we ultimately selected, they were very difficult to escape with single nucleotide. So we chose them based on the difficulty by which they could be escaped in part, not only their potency. At the time, Mike Diamond did human ACE2 transduced mice, and we showed they worked in mice, both weight loss and virology and histopathology. And then Dan Baruch worked with us in macaques, and we didn't see a single molecule of RNA, genomic RNA, newly made RNA, animals treated even with these monotherapies. So these kind of potencies give you a fantastic in vivo efficacy. So for 2196 and 2130 that ultimately became these drug names that were in Nevis-Sheld, we actually did the discovery in 25 days before we transferred the sequences. So we went from sample to giving AZ the sequences in 25 days. So the speed with which we can do these, not just the scale, is just blows your mind these days. And we could go faster than this now, but I mean, who needs faster? So then, now I'm getting to, I know Joe and Lisa have mentioned we're going to talk about FC media activity. So is it good or bad or indifferent? And we work with Mike Diamond early on with some of these representative COVID antibodies, and this is actually from the Cell paper. You can go back and look. But the point being, you can look at prevention and early therapy and late therapy as different stages and actually different compartments probably that you're dealing with where the virus is. And in prevention, we found the neutralizing activity is sufficient. And how do you do that? Well, you take an antibody, this one's called 2050, and you make a matched molecule that has LALA-PG, which are multiple mutations in the FC region where you knock out activity. And people have studied LALA before. We find adding the PG to LALA is a better knockout for rodents because you had the incomplete knockout with LALA. So we use LALA-PG as the full knockout for rodents. And basically, you can compare the wild type antibody and the LALA-PG, and you don't see any difference in prophylaxis. They look identical. So for prophylaxis, it's pretty clear neutralizing activity is fine, and you don't even probably use FC mediated activities. Now, in Mike's mouse model, if you then transition, this is now therapeutic activity, and you wait, you inoculate, you infect the mouse, and then you come back and you're using a 2050 or a LALA-PG variant. Now you see that you have weight loss with the LALA-PG, but not with the wild type. So you see a different need to bring in the FC mediated in the therapeutic window versus prophylactic. And of course, people are moving from not yet exposed, early infection, moderate infection, hospitalized, and ICU. So this is not an either or prophylaxis or treatment. This is sort of a spectrum. And so then the question is, can you correlate any of this animal stuff to human setting? And I'm skeptical. I'm going to show you what I think are examples from what happened in COVID, because this is where we've seen a comparison that was not a fully controlled comparison, but in the clinic, multiple molecules went forward. So this is bamlanivimab, which was a Lilly antibody, the first to the clinic. Amazing job. They got this stuff in people by June of 2020, probably, really historical event. But if you look at the New England Journal paper of the hospitalized patients, the percentage of patients with an adverse event was 19% in treated and 14% in placebo, which was considered non-significant. So basically they said it didn't work, but it didn't hurt anybody. It was 19 AEs versus 14. But I went back to the preprint, and this is from the preprint, if you look when they segregated those who were neutralizing antibody positive in their serum, the time to death flipped from antibody helping you to antibody hurting you if you're already seropositive, meaning you're farther along. Now the p-values here are 0.18 and 0.11, so they're not significant. But if you actually look, compare the hazard ratios with and without antibody, they're PO3 and PO4. So I'm not saying, I'm raising the question, were there increased death in SAEs in seropositive patients who were treated with BAM? That was an antibody that had a wild type FC. If you look at other antibodies that were taken in the ACTIV 3 NIH-sponsored trials, strovimab and the Brie antibodies, there was basically no effect. You don't see a beneficial effect nor an adverse effect, and those studies were published in Lancet. The only antibody that went forward that was an FC knockout, which was the AZ version of FC knockout, they call triple mutant. There's 3 mutations in the FC that abrogate FC function. Those molecules were complicated because they were YTE to be long half-life. They were 90 days or more half-life. They were also the triple mutant. And they worked in prophylaxis. If they were given within 3 days, they worked in outpatient treatment, and the mortality was 30% lower in the clinic. Now, the primary outcome was time to recovery. So in essence, the trial failed. But if you look at mortality, the only antibody that worked in hospitalized patients to reduce mortality was the one antibody that was knocked out for FC function. And this is why I'm not saying this proves it. I'm saying it raises the question. You could have an antibody that increases your hazard ratio for death or SAE. You have some that are neutral, and the one that was knocked out gave you 30% reduction in mortality. I think it raises questions, what is desirable during therapy? And then finally, Lisa mentioned, we're not only working with Ebola and SARS-CoV-2, we have started a program called AHEAD 100, which is advanced human epidemic antibody defenses. And we're working on 100 antibodies or targets because we think antibodies are really important. So I think not only vaccines for prototype pathogens, but we need to do this for antibodies as well. And I don't think people would argue. If there's something that's not on this list and you love it, put my email right here, especially if you've had it, email me and we'll work on it. So in summary, the speed of making antibodies, especially potent neutralizing antibodies, is super fast now. Long half-life allows antibodies to be preventative, not only treatment. I didn't mention, but we can give antibodies IM now, which opens up a lot of possibilities. You can get to the clinic rapidly, and I think we should prepare ahead of time, not always scramble. And I think antibodies are increasingly the future of preventing and treating infectious diseases. And then, back to the core concepts, what type of antibody? And I'm advocating start with neutralization, get the very most potent antibodies you can isolate. Ideally something about 1 nanogram per ml IC50 if you can, and then ask, do you want to add or remove FC-mediated effector function, depending on the disease that you're treating and the state of the disease. Are you trying to prevent, or are you treating severe disease? And you might have different answers whether you do or don't want FC, depending on the state. And I just want to mention my group. This is a picture of our group who worked on many of these topics. And then the collaborators, I mentioned Mike Diamond, Dan Baruch, Jesse Bloom. The AZ team was led by Mark Esser, and many, many others. And the Ebola work we've done with Tom Geisbert and Alex Bekreyev at UTMB. A lot of companies we worked with. A lot of people gave us money, especially the NIH and DARPA, but including our celebrity locally, Dolly Parton, gave us the first money for COVID and really helped us get started. So thank you for listening to my kind of ranty talk. Wanted to be a little bit provocative for this group to, bring up neutralization vis-a-vis FC, and I'm happy to, take any contradictions of that idea. I'm sure there will be some. So thank you.
Thanks, Jim. That was great. That's the reason that we've invited you. You said, I don't know why you've invited me, but the reason is because we want all opinions, controversial, whatever it might be, so that we really get a holistic picture of effector function. There's a few questions in the chat. Joe, do you want to go through them, or you want me to?
Sure, we can we can go through them. And actually, I guess one of the, I'll start with one, maybe we can switch off. But Mika Schmidt has a question that actually falls off of the last slide that you presented, which was directed to what you said was, get this as potent as you can be. And I guess the balance is always potency versus specificity. And so Mika asks, broadly how broadly are the ultra potent monoclonal antibodies neutralizing against subvariants? And are the epitopes that make it ultra potent also the ones that are well conserved, or is there it seemed to be a direction there? So
yeah, that's a great question. So historically, I think HIV was sort of the form first of this. You'd get really potent antibodies, but they'd be strain specific, and people sort of will never see broad and potent ones. But then, the IAVI group went out to Central Africa and collected people that weren't treated, who had high neutralization titers. And you could see people who had enormous breadth and find antibodies, these PGT series that were broad and potent. And I think, to me, that was sort of a clue. If you can find conserved places on HIV with all the glycans and so on, there's probably the possibility that you can get broad and potent for most viruses. So specifically about SARS-CoV-2, we've been doing recent campaigns with people who had, Omicron variants, and we have antibodies at XBB, BQ1.1, and everything. So, if the question is, can you get broad and potent antibodies for COVID? The answer is absolutely. They're there. But if you're making the point that those are rare and usually the most potent are going to be strain specific, that's also true. So, that's why I'm excited about these ultra high throughput techniques, because if it's 1 in 10,000, it used to be we would never see those and say they didn't occur. Now we're always going to see those because we know what to look for. I think this ought to be something we're always looking for. So if we're going after a flavivirus, we should be thinking pan-flavi. Why don't we solve more than one virus at a time rather than only focusing on the one at hand? So beyond variants, but looking for pan-family antibodies. I think there's potential there. So that's a great question. But I agree with you. There's usually a trade-off for most antibodies, potency versus breadth. That's the typical trade-off, but we're looking for these rare things.
Do you want to take the next one, Lisa? Take your pick.
There was an interesting one that actually came through as you started, you showed your sort of laundry list of things as well in terms of the monoclonals you've sort of got in your freezer. But this came from, I'm not quite sure who it came from, it's anonymous, but what do you think about effector function as the primary driver of the effect for bacterial targeting mAbs? Are we better off targeting toxins versus the bacteria itself?
Well, that's interesting. So, I live in a top 10 medical center, and people are always saying, hey, can you make an antibody to my thing? And it's usually cancer, and we usually say no. But some bacteriologists have tricked us into working on bacteria, and we've worked on Acinetobacter, Klebsiella, stuff like that, and done a lot of work on staph, pertussis, and so on. And I think, yeah. And what I would, to be honest, working on bacteria is pretty frustrating in my experience, if you seek surface proteins, because bacteria tend to have redundant adhesion factors. And so you can cover up 2 or 3 of them, and the bacteria still have 3 or 4 more they can deal with. And they have different phases, and depending on, are they in a biofilm or not? Bacteria are very difficult targets for single target interventions. So I agree. I think bacteria are probably have a whole different set of rules. Now, toxins, on the other hand, we have been working on pertussis and some of the staph toxins. You can find fabulous antibodies to toxins. So depends on how many toxins are involved in the disease. So for staph, people argue about that, but the bugs are putting off a dozen toxins. So if you eliminate one, is that enough to give you clinical benefit? And I'm not convinced. So it's easier to make really good antibodies to toxins than it is to surface proteins. But it's sort of how many toxins do you have to target? And
there
you can
could be-
extend this to things like snakes and scorpions and spiders. And so some of those things have one toxin in the venom, and some of them have 100 toxins in the venom. So you gotta understand the disease pathogenesis before you launch out willy-nilly making antibodies. That's what I would say about that.
And I guess when it comes to bacteria, depending on your indication, there could be different toxins or, site of infection. So you might have to think about matching indication to, the actual protein itself as well, which is interesting.
Yes, for sure.
All right. So there's another question from Corey Momont. Do you think there's a correlation between the antibody affinity and the ability of the virus to escape?
In general, that's true. The difficulty in that is how are you measuring affinity? So I would say categorically, in general, most recombinant proteins that are used for virus work do not fully recapitulate the appearance of that target on a virus surface. Some are pretty close, but if you make a soluble protein, it's probably not identical to the virus. So, are you studying affinity to a recombinant protein that's slightly conformationally relaxed? Is it a monomeric protein or is it oligomeric as it should be? Are the glycans of the cell you made it in the same as the human airway cells or whatever the target is? The glycans might be different in your protein. So did you make it CHO 293 or whatever? So there's a lot of caveats. So in general, I'm biased against proteins. We use them all the time because they're nice and easy, but you're probably better off using transfected cells as your target, or even infected cells or infected primary cells, or, how close can you get, organoid, how close can you get to a human being infected tissue? So the closer you are to a tissue when you're measuring affinity, the closer you will be, well, not even tissue, but virion particles. So virion particles are often awesome. And now we're back to, why don't we just screen for neutralization instead of affinity? That's kind of, our lab just keeps coming back to binding assays really are problematic. And why don't we just screen with the end in mind, which is neutralization? So that's kind of our loop. We try to get out of affinity and binding and get toward function of neutralization.
And avidity.
But yes.
Could be really, we haven't talked about
avidity as well. I mean, there's a whole world of discussions about affinity, and affinity can be broken down to on rates and off rates and steady state, and that's epitope specific, whether you need on and off. That's a pretty complicated area.
Awesome. All right. And Mike is on, Mike Diamond, and asked in the
my
context.
goodness.
Hi, Mike. In the context of variant viruses, some mAbs lose neutralizing activity before FC effector functions due to differential requirements for stoichiometry of binding. Why not optimize both to minimize the issue of resistance?
Yeah, that's a good question. So I think what Mike's saying is you may still see FC effector functions with reduced binding. Neutralization is significantly reduced. And that fits with what I sort of said at the top of the talk, which is if you're studying antibodies of low neutralizing potency, then the relative contribution of FC mediated becomes much more important. It may be matched. I mean, there's even instances where you have a non-neutralizing antibody that mediates protection, and in that setting, FC becomes the only or the dominant force. So that's true. If you start with a neutralizing antibody and you have reduced or no neutralizing function, but you still have some binding, yes, then FC effector function compensates for that loss. And so, Mike's question is, well, why don't you just have both? And I think that's probably true, unless back to there's evidence in flu sometimes where you throw an antibody into an infected lung and dump a lot of complement fixation on that lung. At that moment, that's not good for you. You don't want that. You want the antiviral effect without complement, without other effector cells. You just want, you already have effector cells there. You already have complement. You don't want to enhance that. So I think there's some settings where you don't want it, but sure, a lot of settings where the effector function may compensate for the loss of activity. So yes, Mike's correct about that.
And we did bring Mike up to the panel. So if he didn't hit it, we've given him a space to actually ask the question again.
Yeah, let him contradict me. That's a good
Jim, I'm not contradicting, but I would say that thinking about viruses and thinking about the possibility of viruses to evolve, for those viruses that are sort of relatively, tighter, if you will, don't have a lot of ability to really change as much. I totally agree. Then you can just focus completely on neutralization, find ones that you define as broad, and probably you don't need FC effector function, and you avoid this question that you're raising of potential theoretical concerns. The issue becomes for viruses that can really change pretty quickly. And even though you've selected them for breadth, what we have seen, and as we've seen with Evusheld, even though you had two great candidates, they worked so far and then they died against BQ11 and the others. And now we have to go to the next generation. But if you had FC effector functions in those and they still bound a bit, they might still have efficacy as you go forward, although they would maybe not as much, but they still might show efficacy. So I think you have to sort of think about it in the context of how variable the viral substrate is over time when we think about should you retain it. Forget about maybe optimize wasn't the good word, even just retain. There's optimization, you're right, that's going into a different question and there could be lots of issues associated, but just retain the natural ones I think may be beneficial in that context. Over.
Yeah, and Dr. Schmidt's asking the same question. What happens when, high neutralizing antibodies at low concentration late? And that's true. If you're neutralizing tighter in the person, falls sub-threshold for protection, and you still had some FC around, yes, it might help you. I wouldn't contradict that.
Yeah, it's an interesting thing to think about, especially in the context of COVID, right? I mean, we don't have the sort of broad epitopes that we see with things like flu yet. I mean, there have been identified epitopes, and actually Nancy Hegwood is on, who also identified some of these antibodies to broad epitopes, but she's asking about natural human mAbs versus bispecifics or trispecifics. And I know Nancy just had a publication on this. So what are your thoughts around that?
Okay, so I can be ranty here too. I grew up as a pediatrician, infectious disease person, and I have sort of a first do no harm hat that precedes my isn't our biotechnology really cool hat. And so I personally prefer that we use monoclonal antibodies that are as close to something that occurred in a real human being as possible. If there's no other choice, like you're dealing with cancer, people don't typically make a lot of great human monoclonal antibodies against host proteins that are going to eliminate cancer. So sure, have at it, make nanobodies and all kinds of crazy stuff and make them tri-specific. But if you're going after a virus, typically we have human beings, they make great antibodies. Why don't we use naturally occurring antibodies? Because you're less, the farther you get away from a natural antibody, naturally occurring antibody, the less predictable your off-target effects are going to be, your tissue cross-reactivity, your anti-ID problems, seeing that engineered antibody is foreign. I'm not saying it always occurs. It usually won't occur, but sometimes it will occur. And so your ability to predict that with likelihood gets less and less the more engineered it is. And I have to say, I got influenced because I was on the SAB at MedImmune when palivizumab, which was their lead product, they made a somatically mutated version of that that was reactive and bound to cotton rat liver, and they re-engineered that out, and they thought they removed all the tissue reactivity, and they went to a phase 3 trial, and they ended up with a bunch of rashes in people and so on. And I would say that was like $1 billion test of, do you care if you engineer an antibody and along the way it had a little tissue cross-reactivity that was non-natural. And I saw that and I said, wow, I never want to do that. I think I would just stick, why not go back and just find a good antibody rather than engineering an antibody? So that's most people don't agree with me about that, but I would say I prefer natural antibodies when they're available, for sure.
Motivizumab was an interesting story. I guess it also depends on how you want to use it. So if you're thinking about prophylaxis versus like a one-time treatment, I mean, motivizumab obviously is an outlier there. You could think differently, but you really have to be careful on your non-clinical experiments if you're going to move that forward.
Yeah.
Right.
And
I
also,
think,
who's your population? Is it pancreatic cancer patients who have nothing left, or is it a normal baby where they've got a life ahead of them? To me, the balance sometimes, you'll do some things in some settings you wouldn't do in others, the risk benefit.
I think we're actually at time, right? We were going to 11. So there was more question in the chat, but we could probably get back to Ha with an answer there by email or something.
Stay tuned. I'll talk about that next time.
All right. Well, thank you so much. This was a great, as expected, fantastic talk. Great discussion afterwards. Appreciate your candor and being a little bit provocative. That is exactly why we have these, to have that. That's why we leave time to talk for questions. So terrific, and thank you very much. And for everyone who joined, thank you very much for hanging on and being part of the discussion. So appreciate it very much. And we will, as it was pointed out, Lisa pointed out early on, we have another one of these coming up on the 21st. We hope to see you all back then. So thanks very much.
Thank you so much, Jim. Thanks, everyone.
Thanks.
Thank you.